Hello, everybody. My name is Abby Green. I am also from Radford University, working in the same lab as Natalie, but I'm going to be sort of showing you the more computational side of our lab. So Natalie does a lot of lab work. I'm more computational on the computer. So sort of different lens of what our lab looks at today. So first, what exactly is macrogenetics? Macrogenetics is where we're able to look at very large scale patterns with lots of different species and sort of see how genetic diversity looks like across a large geographic space. So for example, this project here was looking at phylogeographic breaks in birds across multiple continents and with over 200 different species involved. So this is kind of the large scale analysis we're looking at when we look at macrogenetics. And the cool thing about it is not only do we look at these large scale patterns, we're also able to sort of reuse data from past projects. So people are out there collecting this genetic data all the time, but it gets used, sometimes it just gets used for one purpose, and then it's just sitting in a database. This allows us to reuse that data for these really cool large-scale analyses. And one tool to be able to get this large-scale data is Phylogator. Phylogator is a data aggregator that takes data from GBIF, NCBI, and BOLD and puts it all together, giving us almost ready-to-use georeferenced DNA sequence alignments. So the most important piece of these alignments is that georeferenced piece. So not only do we have these alignments that are almost ready to use, we also know exactly where every single sequence came from, which is really important for these macrogenetic studies to be able to look at these large scale patterns. Because we're looking at such big amounts of data, we use a tool called predictive modeling to be able to analyze it, and sort of what that is we're trying to predict an unknown outcome based on the response of past outcomes or past data. The tool specifically we use for the projects I'm talking about today is random forest analyses. And how this sort of works is it makes these decision trees shown here in this picture. And on the tips of those trees is our response variable. In this case, it's going to be a genetic diversity value. And then within the tree, we have different predictor variables. So these are our geographic, climatic, and life history traits in this case. And sort of when you're trying to predict it, your sample that you're trying to predict something about follows down the tree, making decisions at each of those points based on the predictor variables, and then lands at a response variable value at the bottom. And how random forest works is it not only makes one of these trees, but it makes a whole bunch of these trees, and then we're able to compare them and see which variables are most important, which is kind of what we focus on in our lab, is what variables are most important for predicting those values on the tips, that response variable. So what exactly do the projects I'm going to be talking about today looking at? So we are looking at what geographic, climatic, and life history traits contribute to two different measures of genetic diversity. First, we're going to look at the presence of hidden genetic lineages in salamanders. And then next, we're going to be looking at interspecific diversity, so that within species diversity. So very similar methods, but having a different response variable in that predictive modeling. So first, we're going to focus on the hidden genetic lineage project. This is my specific project that I worked on versus the second one was my lab mate Hannah's work. So let's all get on the same page about what hidden diversity is. Because of the limitations of resources and time that goes into taxonomic work, it's such an intensive process. We haven't necessarily formally described every species that is out there. So the idea is that many of these species that we do have contain hidden diversity. For example, the plethodontid salamanders were often thought to be way fewer species than they are formally described now. So they're a great example of how hidden diversity has now been formally described. So that hidden diversity is our response variable, and I'll talk a little bit more about that in a second. But here's a sort of distribution of where all of our genetic data came from. All of our data was downloaded from that Phylogator database that I mentioned before. We looked at two different loci for this project. We looked at both Site B and CO1. We had about 1,800 sequences involved, and 83 species were represented in our data. So that's sort of a distribution of where all our data is coming from. Just kind of cool to look at. So the first thing we had to do is we used species delimitation to be able to decide which of our species included in our data set contained that hidden diversity. And because there are so many different species limitation methods out there, and they all perform a little differently, whether they're a distance-based method or a tree-based method, they're going to kind of have different results. We wanted to compare multiple methods to make sure we're getting the best answer. And how we went about doing that is we looked at 3 different species limitation methods across both of our loci, and we had to have a way of sort of summarizing the results. So we ended up with 2 different models. We had our all agree model, where we only used that data if every single species limitation method agreed. So in this chart here, you can, each of these rows is one of our species, and then these little boxes are the response of the species limitation method. So if the box is green, that means that the method predicted that there are more than one genetic lineage in that species based on our data. And then if it's red, that means that it predicted that there's only one genetic lineage. So if it's green, it's it contains that hidden diversity. Red, it doesn't contain that hidden diversity based on that specific species delimitation method. So our all agrees model is all of these rows where every single method agreed. And so if every single method agreed, it was included in that all agree model and either labeled as not hidden or hidden. And then for our majority rules, we have more of a best 2 out of 3 sort of situation. So if you look at sort of some of these rows up here where there's sort of a mix of things, we went with whatever answer was most common in that row. So these ones would be labeled as not hidden based on that majority rules model, but they would not be included at all in the all agree model since there was sort of some disagreement there. So the majority rules model allowed us to use all of our different species, whereas we had to leave some out in the all agree model. So that's our response variable for that random forest predictive modeling. Next, we had to curate our list of predictor variables for the predictive modeling. And for this, we had climatic variables from the Chelsea database. We had a handful of variables that had to do with like human influence. We looked at geographic variables, looking at latitude, longitude, that sort of information. And then we also had a couple life history trait variables from the Amphibio database that we sort of filled in some missing gaps with AmphWeb and some other online resources. So those are predictor variables. Those are on the nodes within the trees. And then so results from our random forest analyses, we did 8 different random forest runs. So for each of our majority rules and all agreed models, we also did some correlation cutoffs. So if variables were highly correlated, we removed one of the pair that were highly correlated together to sort of cut down on our list of variables. We had about 120 different predictor variables going into these models. So by doing that correlation cutoff, it helped us reduce that a little bit to when things were highly correlated. Coming out of our random forest runs, all of them had really high model accuracy, but only one of them was actually significant. And the biggest reason for this is that our data was very much skewed towards species that contained hidden diversity. So whenever a model predicts that it contains hidden diversity, it's more likely to be right. So that's kind of why we end up with those high accuracies, but only one of them was actually significant. So moving forward, looking at which variables are most important, we focused on that one significant model. And then here is sort of some results from the random forest about what predictors are most important. With random forest, it gives each variable in the run an importance value that we can compare to see which ones are most important. And so we looked at the top 5 importance values and that were associated with our variables. And while among those top 5 variables, there wasn't a lot of like one wasn't like super more important than the other. The thing that we noticed is that every single one of them is a standard deviation value. So something about variation in climate seems to have an impact when we're trying to predict this hidden diversity in salamanders. So now we're going to kind of change gears a little bit, moving on to the second project. This is the work of my lab mate, Hannah. I did not work directly on this project, but it's very similar to the first one. We're just going to have a different response variable. So instead of looking at hidden genetic lineage, we are now looking at interspecific diversity values. So they had 3 different interspecific diversity values. They looked at nucleotide diversity, isolation by distance, and isolation by environment. So that's the biggest change between the 2 projects. This is a distribution of their data. They also started with that Phyligator data that we did, but they were able to almost double their data set by doing some manual data curation. And they also only looked at site B. So while we had the 2 different loci in ours, they just had the one. And this is sort of a distribution of their data. This is a distribution of that nucleotide diversity value, so one of their measures of genetic diversity in this case. And as you can see by looking at the map, there aren't a lot of immediate geographic patterns just looking at it. So that just makes it even more exciting to go into the other stuff. This is a lot to look at, but this is sort of the results of all three of their variables. So we have the tree with all of the different species they had in their data set. Those red and black bars are the isolation by distance and isolation by environment variables. So if it's red, it was a yes for that species. If it's black, it was a no for that species. And then these blue bars are nucleotide diversity values. And again, just like with the geographic patterns, we're not seeing any immediate taxonomic patterns either. So that just makes it really exciting to run into the random forest. And here's a little bit of results from their random forest analyses. They ran one random forest for each of their response variables. And so sort of orient yourself on these charts. The x-axis is that importance value that I mentioned when talking about the first project. And then the y value is their top variables for the random forest run. And the biggest thing that came out of all three of these random forest runs is that range size is the top variable in every single one. So something about range size is important when we're looking at intraspecific diversity in salamanders. So to sort of wrap it all up for you, what we found from both these projects is that that choice of diversity measure matters. When we're looking at hidden diversity, we're seeing that variation seems to be important. When we're looking at that intraspecific diversity, we're seeing that range size is important. And if we looked at other measures of diversity, like phylogenetic diversity, for example, which is another common genetic diversity value that we look at in our lab, represented by these little phylogenetic trees down there, we would probably see different variables pop. So when we're looking at this type of analyses, we really want to make sure that we're choosing the right variable for the type of conservation that we want to focus on. And that's sort of the conclusions from both of those projects. And on that note, I want to do a couple of thank yous. We collaborated with other universities on both the projects I talked about today. The first project, we worked with some people out at Ohio State University as well. And then for the second project, we worked with that whole group of people at the University of New Mexico. So thank you to them. We could not have done those projects without them. And then also a quick shout out to NSF for funding the projects and the Radford University Office of Undergraduate Research and Scholarship for making it possible for me and Natalie to be here today. So on that note, does anybody have any questions for me? We have time.
Thank you so much, Abby, and you do have time to take a few questions from the audience.
So when you look at range size, are you looking at the discrete population? Can you that out or
So for that variable, again, that's not the project I work on, but I'm pretty sure what they were looking at there is the range size of like the data they had. So like within each species, how far is it spread out for the data that they had in their analyses? It's just like the range size of that population. Does that make sense?
So you mentioned
you think, or you like, is it high variable climates or low variable climate conditions that result in this?
I'm pretty sure if I remember correctly, it's higher variation, the more diversity, the more likely it was to be hidden diversity. Yeah.
Yeah, unfortunately, this will be the last question. I was curious, was the phylogeny that you were using in the study, was that one that you built with stuff you pulled from Phylogator, or was it like some other amphibian tree that you pruned to match the species that you were able to?
Like for the second project with the tree,
Yeah,
I'm not totally sure.
Yeah,
I did not
I
work on that
your project.
project, so
Okay.
you could ask Dr. Pelletier about that. She'd probably know more than me on that one.
All right, well, thank you so much.
Thank you guys.